Hi, everyone. I would like to welcome you to today's webinar on engineering the future of drug discovery. My name is Lucy Su. I'm a senior product manager at Twist Bioscience. I'm very excited to introduce you to our speaker today. Murat is currently the co-founder and CEO of Antiverse, an AI biologics organization aiming to engineer the future of drug discovery, which he founded alongside CTO Ben Holland in 2017. Murat has a background in software engineering and bioinformatics, where during his mid-career, he developed high-frequency trading systems. After shifting into the biotechnology industry, Murat developed cell imaging software and lab robots to accelerate cancer research. He has also worked with Thermo Fisher Scientific, where he established an automated approach to gene synthesis workflows. In addition, Murat founded Starlight, a consultancy company. Let's get started. Welcome, Murat.
Thank you very much for the introduction, and thank you for everyone being here and listening to us today. I would like to tell you a story about AI in drug discovery, and it's a big story to tell, and I would like to share with you our version of it. We will be mostly focusing on biologics, but we'll also briefly touch on small molecules. I expect this presentation to take about 30 minutes. I'll start with AI in drug discovery in biologics, and then talk a little bit about the obstacles that we see right now and how can we resolve those obstacles to engineer the future of the drug discovery. At the end, and from that point forward, I will give you a couple of examples how we play into that field and maybe a case study to give you, to hopefully leave you with a little bit more information and a few actionable ideas. So where are we when it comes to AI and drug discovery? First of all, a lot of things is happening on the small molecule side because there's more available data. But if you focus on the biologic side, about 10% of the whole pharma pipeline is now AI derived. It doesn't mean all de novo discovery. There is a lot of optimization, or maybe the target is discovered through AI. But one way or another, about 10% of the current pharma pipeline is considered to be AI driven. And you see a couple of examples around COVID, mostly in oncology and maybe one or two in neurology. But how do you think about this space, AI and drug discovery in general? So we mostly split it in two parts, small molecules and larger molecules. And you see most of the AI companies being on the small molecule side. There are two reasons for it. First, the mathematical rules of protein-protein interaction is better studied than antigen-antibody interactions. And the other advantage of being in the small molecule area is the amount of data and also how easily you can generate more data, how easily you can run your validations. So if you shift your focus to biologics, which might be preferable for a couple of different reasons, specificity being one, then you see companies, again, either focusing on de novo discovery or optimization. And within the optimization, there are different parts, affinity maturation, developability, solubility, etc. And when you look at the de novo discovery part, then there are only a few names there, us being one. And within that sphere, again, you can divide them into two, one of them with pure in silico approaches and the other ones that are using wet lab technologies to almost marry that technology and build something together. Both have advantages, and I will just move us maybe to the next level where we see the obstacles. First of all, data quantity. When it comes to antibodies, if you look at the PDB files, there are maybe thousands, but not 10,000 or 100,000 VHH antibodies, and especially the paired ones. The quality, all of the publicly available data is coming from different resources, coming based on different experiments from different labs. So there are certain deviation, and normalization is not that easy. Then there's access. So some of the big pharma and biotech companies have this type of data generated in the past, but not necessarily true for all of them. And again, it might be completely in a distributed fashion, staying in somebody's hard disk, living in an Excel file somewhere else, and so on. So even if you have a collaboration in place, accessing that data may not be that easy. And then if you decide to go down the path to generate your own data, you can do that. You can run certain experiments that takes a couple of weeks to a couple of months to generate data, and we eventually decided to do that where everything else failed. But it's definitely a barrier to entry to the market. Then there's the speed of generation. So it doesn't matter how much money you have, you will definitely hit certain, it's not going to be a simple, some application of AI in another field where you can just generate data. So the second part, the second biggest problem we see is the validation. And by that, I mean the validation of the predicted molecule, whether you have to synthesize it, express it, purify it, or you have to check whether it binds and has correct characteristics. That takes time. There is no real high throughput method. There are some companies that maybe does monoclonal antibody expression purification, tens or hundreds level, but we haven't seen anything that does it on the thousands or 100,000s level. And then another one is size of biologics, but it comes with the area. If you think about a small molecule, it might be 200 Dalton, where on the antibodies, 150 kilodalton and so on. And then there's the complexity. I use the word antibody sometimes instead of biologics, but what I mean by that, anything with a heavy chain. We have VHAs, multispecifics, PROTACs, intrabodies, and so on. So there is a huge amount of complex to be modeled. Now, some of them also fit into the future possibilities because we see more and more improvement in this field with writing and DNA is getting cheaper and faster. You see that you can maybe synthesize some molecules even with a desktop machine. Lots of high throughput experimental models being developed both in big pharma but also in academia. New modalities are emerging, so there will be multiple new ways of targeting not single drugs, targets, but various targets in combination by using multispecifics and designing those multispecifics hopefully going to be easier. We are hoping that AI will offer some information on designing the best VHAs that can cross the blood-brain barrier. And if you extend it a little bit further, I think what we will see and what we are already seeing an increase in the percentage of AI-driven or AI-native molecules in the biologics pipeline of big pharma. And I think if you stretch that quite some far, hopefully we will get to a day where we will have true personalized medicine. It's very difficult to guess, I think, when that will happen. But you see that you can get an FDA approval now on software and sorted devices, mostly for diagnostics, but I'm hoping for a future where it will happen for a therapeutic as well. So you get an approval on your algorithm rather than each individual asset that you generate for a particular person. Now, we play in that space by mostly focusing on not incremental updates, efficiencies, making optimization faster, affinity maturation faster, that kind of stuff. We would like to do something that wasn't possible without AI before. And that is going after GPCRs and ion channels, challenging targets that have been maybe successfully drugged with small molecules, but not so much with antibodies. There are only 2 antibodies approved by the FDA against these GPCRs, but there are hundreds of them, and they are associated with tons of different diseases. And we believe that this is really a good area to highlight what AI can do. Now, there are two problems when it comes to that, and I'll talk more about it later. So, what are we doing? We are using AI, but also we are using a lot of wet lab innovation to generate de novo antibodies against GPCRs. Our mission is to go after cancer, cardiovascular disease, diabetes, and a few others, all associated with these GPCRs. We are a team of 19. So here's a little bit about Antiverse. We are a team of 19. We found the company back in 2017, raised about $5 million. We generate our own data. So we are data independent, and we use that data to build machine learning models. Then when you point that to a new target, it starts predicting new antibody sequences. Then we turn into, with the help of the WISP, into an antibody library. We get that library, screen against the GPCR, and hopefully bind some target-specific binders. Hopefully they also have function. Now, we work with about 6 different companies so far, 2 of them being after pharma. Now, when you go after GPCR, there's a reason why we have only 2 FDA-approved antibodies against GPCRs as an entire industry, because there are 2 big problems. One of them, we call this a forest problem, that GPCR, which is a multipass membrane, transmembrane protein, it is hidden among a forest of other proteins. So when you try to bind an antibody against it, it's very well hidden among many other things. Now, the second problem is an iceberg problem. So this is somewhat of a big protein, a receptor, GPCR, but most of it is intracellular. You have just a tiny bit on the surface. So your effective epitope space is very, very limited. We have 2 solutions to this. One of them is we have some methods to make sure that we have high expressing cell lines, basically, that have the 1,000 times more GPCRs or ion channels on them, that increases your chance of hitting one. This is a inducible cell line that has a maximum number of receptors on day 2. Then we do our panning on day 3, they die. Then for the second time, we get a fresh batch. The second problem, the iceberg problem, we solve it with the help of AI. What we do, we provide the input sequence of the targets, and based on that, our model designs a target-specific library. It makes some predictions of how well certain sequences will bind. Then we get those, we select a couple of different backbones, and then we send this for synthesis. We receive the DNA back, put into the plasmid, subclone, put into phage, do the panning. So here how that process works in more detail, from target sequence, ideally with structure if it is available, to move to stable cell line development and target-specific library design. That's the point where we work with Twist. Once we receive the library back in our labs, we do the phage display, either against a usual, soluble protein, or it's mostly a biopanning with flow cytometer against the GPCR. Then we send our results for sequencing so that we can not only do random colony picking, but an AI-based analysis of the whole population and make some informed decisions about to select diverse sequences. And I have a nice visualization of that in a few slides. Once we do that analysis after stage 5, we do the expression of those scFVs or FABs or MABs, whichever is the most used, either the preferred choice of structure for the partner. Then we confirm the binding again, and after that we send them for functional testing. That part we do not do in-house. However, there are a couple of very good companies out there, both in UK, in the US, also in Belgium, that does the CM, beta-arrestin, IP1 based assays. And we get the results, and that's what we hand over to our partner. So they come to us with a problem of we have a GPCR, we cannot find a functional antibody against it, and mostly it's an antagonist. And then after 6 months later, we give them a functional antibody for them to decide whether to license it or not. An example of that work is against the GPCR that we have done recently. We got a library from Twist. We screened that library against its GPCR, and we also use a target-specific library as well. And at the end, we had about 55 binders. We got these 55 binders. If we check them, whether they also bind to the transiently transfected cell and a few other things, just to make sure that there is sufficient specificity. And if you can see it, there are a few of them on the right-hand side, and you see the controls on the lower bottom. Now, the next thing we do, we have screened these are all monoclonal antibodies. We screen them both as an agonist and then an antagonist. Bear in mind that these were not designed to be agonist or antagonist. It was a pure screen, so we haven't predicted any function. None of them had an agonistic function, but some of them had antagonistic function, which was really good in this particular case because there are a few different indications associated to that GPCR, thrombosis, triple negative breast cancer being two. So we did IC50 determination, and the affinity levels were not that bad either. Now we repeated that data. We also look for beta-arrestin, which I don't have in the presentation, that confirms also inhibition, antagonism. And then last week, we sent some of those antibodies for a platelet aggregation inhibition test, and we got really good, encouraging results for antibodies better than a commercially available antibody. And one of them is even better than a pharmaceutical drug. So we are really happy with the news that these are not just antibodies for tests or for diagnosis, but that you can really turn them into therapeutic antibodies. Of course, still a long road ahead, but I think for a lot of people who judge the quality of a platform based on how far an asset is, we were really happy to hear the news. We did a lot of improvement to our models. We started with actually sequence-based prediction, but eventually to improve the accuracy to a point where we can maybe skip the whole library design and panning and get to maybe an accuracy of 1 in 10, 1 in 100, so that you can directly synthesize those antibodies or express querify them and then test them against your target. You need a little bit more to go on with than just sequence. So we developed an in-house antibody structure prediction. It is almost as accurate as AlphaFold, if not better in multiple cases. But there are other tools that seems to be better than AlphaFold as well, like OmegaFold or a bodybuilder that are more specific to antibody structures. However, what we optimize for is the speed, because machine learning models can deal with a lot of noise, but you need a large volume of data for most of the applications. So that's what we focus on here. So on the right-hand side, you see a couple of examples of the ground truth in blue and the prediction in green. And the RMSD is usually under 1 angstrom in most of the cases, especially around HCDR3, you see that increasing between 1.5 to 2.5. So this is the slide I wanted to talk about. So this is a presentation of a panning campaign we did, a discovery campaign we did for a big pharma. Each dot here represents an antibody. If you think about it, so this is coming all from PAN3. It's a 2-dimensional analysis of that data. If you include the color, it's 3-dimensional. So each color block is is similar to each other, both sequence-wise and predicted structure-wise. So it is clustered actually in a 20-dimensional space, but then for visualization purposes, it is squeezed down. And how is this helpful? You would expect that similar sequences to bind the same epitope. So if you have a limited budget, or if you don't know where the agonistic or antagonistic function really is, and you want to have a lot of epitope coverage, what you would do in this particular case, you would go to each different color, flamingo, blue, purple, yellow, and so on, and you would pick a couple of sequences, representative sequences, and we would give that to our partner. Partner would then go and test them, and maybe, let's say, from the blue cluster, you would get an antibody back with the desired function, but maybe not all of the characteristics right. So we would do a second run of selection from that cluster and give them and have an iterative approach with the partner till they get what they want. I think that's most of what I want to talk about, but I will briefly touch upon the leadership team. To enable what we wanted to do, we had to have a very balanced machine learning and a wet lab team. So on the machine learning side, it is my co-founder, Ben Holland, graduate from Oxford University, did a lot of interesting stuff, being in Rwanda, doing field testing, Malaysia, building a manufacturing pipeline, Japan for business development. But he's a scientist and engineer at heart. So he worked on similar problems for a very long time, and we met in 2017 to create Antiverse. On the specifically GPCRs and ion channels, when it comes to expertise, Gersi Hussein is one of the world-known scientists. He spent 20 plus years to develop hundreds of cell lines, and did again hundreds of experiments against those cell lines with VHAs and SCFEs successfully when he was the CSO in Theranix. We are really lucky to have him as our antibody discovery director. Then we got our first funding from Deep Science Ventures, a London-based accelerator, and Kirsten Papanfas is representing them, and some other investors help us with target selection and with the direction of the company. Then we have Peter Pack, who raised about 120 million in total in different companies. Some of them might already know him from his role at Crescendo Biologics as a chairman first, a CEO later. He did, I think, their last round as well, which is quite high. And as we move closer to positioning company as a AI pharma, we need to know more about the indications, how the targets are associated with indications, and what is the commercial potential, what's the competitive landscape, what are other molecules, both in biologics, but also in small molecules. And to cover that expertise, we brought in David Chow. He changed our all internal targets and pipeline in just a few months. So we're really lucky to have him. With that, I want to thank you all for listening.
Thank you, Mara, for a great presentation. We are now going to open it up for questions. So if you have a question, please make sure to ask it in the Q&A box. So first question that we have is, on slide 11, was the model/ground truth overlay a prospective prediction, or was it benchmarking against a known crystal structure?
Yes, it was a crystal structure. But we have more examples of it, so I can share if there's interest.
Thank you. Next question that we have is, at what stages does Antiverse implement AI and ML? And do you plan to implement more computational methods throughout your discovery pipeline?
So we use AI ML for two parts. One of them is the library design, or the prediction of the sequences which we will think will bind to the target. And the second time we use AI/ML is the selection of the sequences that will be tested. So that's the end of the wet lab experiment. And sorry, what was the rest of the question?
Do you plan to implement more computational methods throughout the discovery pipeline?
There is definitely a lot of interaction between the wet lab, the ML team, and we see that there is some tooling necessary to make the process more streamlined. And then there's another aspect of it. So it's not really that black and white when you categorize companies. Oh, these are doing de novo discovery and these are doing optimization. I think everybody starts from some point and move other direction. So for us, it just makes sense to go downstream and start predicting affinity, developability, and so on.
Another question we have is, where do you get your data to do the ML training on?
We initially got it from PDB and OAS, Observable Antibody Space, and a few patents. After that, we started to work with partners, and then they provided some of the data. But I would say the majority of the data we had to create it. We got off-the-shelf targets. We purchased some antibody libraries, and also we designed some. Then we screened those libraries against the targets. Did the NGS, put into a database, and that's the bulk of what we do.
What are the sizes of your computationally designed libraries?
It's a good question. Then there are a few different ways to answer it. I think the shortest answer is about a few 100 million was the first one we designed. And then the second one we designed was about 300 times smaller than that. So the limitation is actually a partner's ability to be incorporate that much diversity in the synthesis system. So usually, I mean, Twist maybe should answer that, but you can only synthesize so many different regions, HCDR3, HCDR2 regions, and recombine them later on.
Do you use a fixed framework, or do you vary your frameworks within the library?
I think varying the frameworks is a good idea, but it's not very quick, or it's definitely that's what we have done originally, but then we settled on a couple of different frameworks where we could just maybe get the HCDR3 synthesized and drop in subcloning to speed up the whole process. And if you look at the VLZ especially, there is a lot of you can do there to reduce the diversity and focus more on the VH.
Another question that followed up with this is there a relationship between the best framework and the specific GPCR?
It's interesting. This is not something we have studied. I think probably that needs to be answered experimentally, because most of the data we have generated is not coming from a lot of GPCRs anyway, for me to be able to say that. But I would maybe expect that to be so.
And do you benchmark against AlphaFold? And do you work with creating VHH libraries? I think the second one might be an incomplete question, but do you benchmark against AlphaFold?
So that part is, in terms of the antibody structure prediction, yes. In terms of the binding accuracy, like how many sequences you need to generate to find a binder, that's not necessarily covered by AlphaFold. And it's two questions, not incomplete.
Sorry, I must have misread it, because the question following that was, do you work with creating VHH libraries?
Yes, we do. Yeah. So I say we work with antibodies, but we work with anything with a heavy chain. I mean, VHH is obviously favorable for a lot of different reasons. One of them being its small size. And also there are a lot of available data sources if you partner with someone who has their own and so on.
What kind of ML model do you use for CDR generation?
What kind of ML model do we use for the CDR generation? It is actually an ensemble of different models. So we experimented variable autoencoders, GANs, graph neural networks. So pretty much all of them.
And another question came in. Could you elaborate a bit on your phage display file panning strategies?
Sure. It is, I mean, there's a lot of know-how there, but on a very high level, it's not much different than the phage display file panning strategies you would get somewhere else. You usually start with a cell line. If you have something to deselect against, then you do your deselection, and then you do a couple of more runs. If there's cross-reactivity in play, then you will try to also search for cross-reactivity too. It usually takes about 2 weeks, maybe less than that, to do 3 rounds of panning. And at the end of each round of panning, you would usually look for diversity to know when to stop, whether to go a little bit more or not. I mean, we do certain things to maybe keep the diversity as high as possible, which is a little bit unnatural compared to traditional way of running phage display.
So how do you choose which to test from the first round, and how many do you test from the first round?
Which to test from the first round? That is completely a budget issue than anything else, right? If you are testing maps and you start with SCFPs, for example, conversion to maps, 96 at a panel will set you back about 60K. So it depends how much money and time you have.
I know that's always the situation. So, and then some other questions that we have is, after clustering, how do you select which antibodies to test? So I guess that kind of follows the question that we asked before.
After clustering, yes. So we usually have not only a binding prediction like binary, but there is a binding score that we can use. Also, I mean, there are things that you look for: the number of counts, and the number of other sequences, the counts of the similar sequences to that particular representative sequence, and so on. You also can look for, you don't have to, the enrichment profile, like how many times that sequences were there in the previous rounds and how did it evolve over time. And there are a few other things.
I see. A question came in to ask if you are able to elaborate on what kind of collaboration models that Antiverse has, fee for service. So.
Usually we go after very difficult targets with big pharma, and it's usually a target that they work on it and they didn't get the result they were looking for. So for that kind of breakthrough antibodies, we usually get milestones and royalties for that, risk revenue share. For other times, doesn't matter how many case studies we have internally, a pharmaceutical partner or biotech partner might be asking for their own proof. They want to be based on their test project. So there were a couple of exceptional cases where we run a fee-for-service POC project. And when it comes to academic partnerships, I mean, we built this company to bring as many drugs to patients as possible. So we are quite flexible in the way how we run the company. So,
and maybe we'll end with this last comment and question. Antiverse, anything specific to the name of your company? Great presentation. Thank you, Murat. All the best to you and your team.
So the founder of the name is actually a very good colleague of ours. He has his own company and he's part of the DSV cohort. He came up with an idea of antibody and universe and combined it as an antiverse to pick the right antibody from the universe of antibodies. And most of the slide deck, you see, these dots around, or the design of galaxies, like antibody galaxies. So we play with that team.
Amazing. Thank you so much. Well, I think that's the last question that we're answering for today. For the following questions, I see that there's a few folks who sent their names, so we'll follow up with you afterwards. And thank you again, Murat, for a wonderful presentation. And thank you everyone for coming. Have a great day. Take care, everyone.